It's a pleasure to present our contribution to Living Data 2025. We really believe that unified information drives transformation. In this regard, we present a critical analysis of environmental characterization practices through integrated analysis of biodiversity data published in an open access database. First, I'd like to present me. I'm Paula Castelões, a Brazilian biologist with more than 20 years acting as an environmental consultant in all Brazilian offshore sedimentary basins, developing innovative projects to understand and protect biodiversity, and building and managing databanks. With me in the present effort were Drs. Luciana Frazão e Cristine Brown Moraes, phytoplankton specialists and also environmental consultants, and Leonardo Marques da Cruz, oceanographer, director of CLS Brazil. First of all, we need to recognize the context in which we live today and critically analyze if what we are doing today is aligned with the future we want to live in or live for future generations. Thus, the efforts made so far with environmental baseline studies, for example, generate all the information we need to address climate changes or conserve, restore marine biodiversity? Looking forward, do we have a wide comprehension of biological carbon pump in the oceans, considering it is strongly influenced by the structure of pelagic planktonic communities? Are the efforts being implemented sufficient to support understanding of the carbon cycle in deep sea? Our concepts may not longer be up to date. But how can we know that? Are there clues about this gap? Where? We will not be able to do anything like this if we assume that environmental follows pre-identified patterns that doesn't deserve or need to be questioned. In this work, we question about the distribution of the microphytoplankton community in the water column. The common sense says that light, the photic layer, restricts their occurrence. Is it true? Clues can be found in standardized open databanks. In these databases, we can find valuable efforts that goes beyond commonly accepted standards. These additionals can justify a careful review of the established patterns. In Brazil, we have an environmental characterization database, BDCA, built to make publish raw data from baseline studies and environmental monitoring projects, conducted under environmental licensing processes with IBAMA, our environmental permitting agency. Nowadays, there are 17 EBS published in BDCA. Among them, only 4 sampled and analyzed microphytoplankton below the photic layer at all water masses of water column, while the other studies covered almost the entire Brazilian coast. These 4 are restricted to the southwestern Atlantic coast, as shown in this picture. Data available in BDCA clearly show microphytoplankton cells naturally occurring below the photic layer. And although their density decreases with depth, they remain diverse. We can show in these 2 graphics, the first one for density, that decrease in the below the photic layer, but remain diverse. Diatoms, dinoflagellates, and cyanobacteria are the best represented groups in the deep ocean, considering both richness, number of taxa, and cells density per major group. Although density decreases with depth, as you can see in the graphics, taxonomic diversity does not necessarily decrease. It remains stable. The diatoms taxa that appeared most frequently in the EBSs were Chetoceros, Cylindrotheca closterium, Pseudonyschia delicatissima, and Thalassionema nichoides. Liu and collaborators also observed this taxa in the eastern Indian Ocean. The presence of healthy photosynthetic cells, dominated by diatoms and dinoflagellates, was observed at depths greater than 200 meters, down to depths of 3,000 meters. These results suggest that fast-sinking mechanisms introduce new organic carbon into the deep ocean. This process is prevalent across the global oligotrophic oceans, as told by August and collaborators 2015. The dinoflagellates taxa that appeared most frequently in EBSs were Heterocapsa, Oxytoxum gladiolus, Protocentrum, and the order Gymnodialis. Liu and collaborators also observed Oxytoxum and the order Gymnodialis at the eastern Indian Ocean at the euphotic zone. The presence of diatoms and dinoflagellates in the deep ocean phytoplankton community suggests the role of the opal ballasting in delivering the particles containing these cells to the deep sea, as recently discussed by Kael and collaborators. The cyanobacteria taxa that appeared most frequently in these EBSs of BDCA were the orders Nostocales and Oscillatorialis. Liu and collaborators also observed cyanobacteria taxa at the eastern Indian Ocean, a photic zone. Ocolitophorids that appears most frequently in these EBSs were Calceosolenia murray, Discosphaera tubifer, Michelsacia elegans, and Ophiaster hydroideus. Although they are not same species, Liu and collaborators also observed Coccolithophorids taxa in the eastern Indian Ocean aphotic zone. The only silicoflagellate taxon that appeared in the EBSs of BDCA below the photic layer was Dictyocha fibula. Liu and collaborators also observed this species in the aphotic layer. Our findings support the hypothesis that fast-sinking processes like opal ballasting can inject fresh organic carbon into the bathypelagic ocean, highlighting the importance of including autotrophic phytoplankton samplings in deep ocean to improve global carbon flux estimates. These results contribute both to a more comprehensive view of deep ocean biodiversity and to their role in biochemical cycles and their ecosystemic services. This time of knowledge construction is only possible thanks to open access environmental reports and data made public through databanks, organized and facilitating systems. By reanalyzing environmental data shared in open databases, it was possible to identify gaps in knowledge of marine biodiversity and ecological services. We hope that it will also be possible to refine global projections and propose improvements in the methodologies and guidelines adopted by decision-making bodies. Thank you a lot for your attention.